for next 10 to 15 minutes, I will present my poster entitled Assessment of the Mechanism for Remdesivir-Associated Clinical ALT Elevations using DILIsym Quantitative Systems Toxicology Modeling. And this poster was presented at the liver meeting last year in 2020. So I'm going to start with a little bit background introduction. So as you might already know, remdesivir gained a lot of public attention as it was granted emergency use authorization in the United States and then approved for the treatment of hospitalized COVID-19 patients. And remdesivir was initially developed for treatment of Ebola. And during a phase 1 clinical study in healthy volunteers, treatment with IV remdesivir, 150 milligram daily for 7 or 14 days, induced reversible low-grade ALT and AST elevations, which are the biomarkers of liver injury. And this happened in a subset of treated healthy volunteers. So in this project, we collaborated with Gilead and performed a quantitative systems toxicology modeling using the DILIsym platform to really understand and investigate the mechanisms underlying the observed liver signals in healthy volunteers. And DILIsym represents the biochemistry and the liver homeostasis and all the mechanisms underlying key the hepatotoxicity mechanisms. And it integrates the drug exposure, often predicted using PBPK modeling, and the mechanistic toxicity parameters, which are derived from in vitro mechanism toxicity assays, and inter-individual variability to predict the liability in human, like clinical situations. And in this project, we developed a PBPK model of remdesivir and its metabolites. And we performed, and actually the assays were performed by Gilead, and the assays were performed, in vitro assays are performed to assess the potential of remdesivir and its metabolites to induce oxidative stress, mitochondrial dysfunction, and inhibition of bile acid transporters. And these are 3 main mechanisms of hepatotoxicity represented within the DILIsym. And so far, using these 3 mechanisms alone or in combination, we have reasonably predicted hepatotoxicity potential of about like 80% of the 82 compounds we tested in the DILIsym so far. And we employed a previously constructed simulated population of normal healthy volunteers that represent inter-individual variability in hepatotoxicity pathways to run simulations at the population level. Now let's look at the results. So starting with the PBPK modeling. So on this left panel, the top diagram shows the structure of the PBPK model we developed. So remdesivir is a prodrug of a nucleoside analog. So after administration, the intravenous administration, it's quickly converted to the nucleoside analog, which is GS441524, and then taken up into the tissue and phosphorylated to form mono, di, or triphosphorylated metabolites. So in our PBPK representation, we focus on the liver because we wanted to better understand the hepatotoxicity. And we lumped all the phosphorylated metabolites as a single chemical entity here. And the plot here shows the comparison of the simulated and clinically observed plasma concentration time profiles of remdesivir and the nucleoside analog, which is a metabolite of remdesivir. So the clinical data from a single dose administration of remdesivir was used to optimize some of the PBPK model parameters. And the comparison is shown on the top panel. And the clinical PK data after multiple dosing protocol was employed to validate our model, like simulations. In the left panel is the plasma remdesivir concentrations simulated and observed. And the right panel shows the plasma concentration of the nucleoside analog. And the red lines represent the simulated profile. And the black symbols represent the mean concentration time profiles from 2 cohorts within this phase 1 study. So overall, the simulated, the AUC and Cmax values were within 25% of the clinical data, indicating that PBPK model reasonably captured the clinical PK data. And in addition, we have employed the acute drug accumulation data of the remdesivir and its metabolites in primary human hepatocytes to inform the liver exposure within the PBPK model. And while the liver exposure from human studies are very hard to get, we were able to use the in vivo monkey data and the simulated liver exposure of remdesivir and its metabolites were generally in agreement with the liver exposure from the in vivo non-human primate data. And now let's focus on the top panel, which shows the summary of the in vitro mechanistic toxicity data. So first, the bile acid transporter inhibition assays were performed using inside-out membrane vesicles for efflux transporters and the transfected cell lines for uptake transporter and TCP. And data indicated that remdesivir inhibits multiple hepatic bile acid transporters, including BSEP, MRP3, MRP4, and uptake transporter and TCP. So inhibition constants were estimated using those experimental data. And data also showed that the metabolites of remdesivir did not inhibit any of these bile acid transporters. And the mitochondrial function was assessed using Seahorse XF analyzer in primary human hepatocytes incubated with remdesivir or its metabolites. And data showed that modest, very mild decrease in oxygen consumption rate, which is mediated by phosphorylated metabolites, indicating that these are mild mitochondrial electron transport chain inhibitors. So the parameter for TC inhibition was estimated using experimental data in the DILIsym platform, which is a standalone software representing the in vitro, the mitochondrial bioenergetics. And oxidative stress was tested using high content screening, and there is no signal detected from the in vitro study. So what's summarized on this table were the mechanistic toxicity inputs used for the simulation. And then DILIsym simulations are performed within DILIsym by combining these in vitro mechanistic inputs and liver exposure predicted from the PBPK modeling. And simulations are performed with multiple dosing regimens in a virtual population, which is about 300 individuals. And the top panel shows the simulated time course of the plasma ALT, which is the biomarker of liver injury. And each line represents a simulated individual. And simulations of the clinically employed dose in the phase 1 clinical trial predicted no ALT elevation. So it just flatline all those simulated individuals overlap because there is no signal predicted. So we ran some dose escalation simulations to assess potential safety margin. Still didn't see anything up to 5x of the clinical dose. And when we simulated like 10x of the actual like phase 1 clinical dose, then we started to see some liver signals, elevations of ALT in a subset of simulated individuals. And the bottom plot is same simulation results, just presented in a different format. Now we are looking at dose and response. And in this case, response is the minimum liver ATP and the peak serum ALT. And again, compared to the untreated group, at 1x, the clinical dose level, ATP and ALT really stays at the baseline. At 5x, liver ATP starts going down, but it's very modest, so it was not really enough to really cause any liver injury. And then it really required 10x of the actual clinical dose to reproduce any ALT elevations. So overall conclusion of this project was that although there were some in vitro signals observed for the remdesivir metabolites for the mechanism of mitochondrial ATC inhibition and bile acid transport inhibition, when we put those together with in vivo exposure in the QST model platform, the simulation suggested that these are not likely to be the mechanisms responsible for the observed clinical trials, indicating potentially alternative mechanisms. And in this project, the analysis was done retrospectively to really look at the mechanisms of the already observed signals. But this type of simulations can be done prospectively for prediction of the first in human outcomes, because the inputs are PBPK modeling, which you can predict first in human exposure, and all the toxicity mechanism data are coming from human-derived in vitro systems. So with that, thanks for your attention. And now I'll be happy to take any questions, well, if we have time.